Any echo? Okay, so welcome everyone for another of our Dunlevie maternal fetal medicine presentations. Before we introduce the speaker, I just wanted to give a few center updates. At the Society for Maternal Fetal Medicine annual meeting last week, we had a great showing, 21 poster and 2 oral presentations. Again, among the very top in the country in terms of number of research presentations at the meeting. And so congratulations to everyone. It is a huge team effort. We, as you know, are doing several searches under the Dunlevie Center umbrella. We have a basic science search ongoing for up to 2 basic science candidates. And we also are in the process of a perinatal epidemiology faculty search. Both these searches are moving along nicely, and candidate presentations have either started or will soon be starting, depending on which of the searches we're talking about. Our next speaker is Dr. Alexander Butwick from OB Anesthesiology, and he will be presenting some of his work on March the 10th. So thank you all. Anna, would you like to present Danielle and her work? And Danielle, if you can share your screen, that would be awesome.
Sure. I'm delighted to introduce Dr. Danielle Panelli. She's our existing WRHR K-12 scholar and is doing wonderful work with looking at maternal stress and activity and how that impacts the obstetric outcomes and pregnancy in general. And Danielle used to be a fellow within the OB or MFM OB division, and it's been just wonderful to see how you continue to grow in your research career. And this is we look forward to your presentation, Danielle.
Thank you so much, Anna, and everybody on this call. Definitely wouldn't be here without all of you. Thank you so much for having me. I think I know many people here, but in case I don't, as Anna mentioned, I am an instructor here in MFM and obstetrics and just completed my fellowship here last year. I don't have any disclosures other than that my time is partially funded by the NIH K-12 that Anna mentioned. So I'm going to start with this analogy that I just want to plant a seed for, both figuratively and literally. I am no gardener, and most of my tomato plants would probably look like the one on the left. But I want to put this here and have everybody think about it, because I think everything that I'm about to talk about, we probably know on a fundamental level to be true, that our environment can really affect the things that we are growing. So keep it in mind. We'll come back to this later. So today during lunch, I am going to talk a little bit about how bad maternal mental health has gotten since COVID pandemic started, talk a little bit about the biology of stress, and bring those 2 things together to put my research on cellular aging and pregnancy into context, talk about how that can affect the baby, and try to end on a positive if we can. So to begin, how bad has 2020 and beyond actually been for mental health? And I think it will come as no surprise to anybody watching that it's been pretty bad. So this is just one example of a study that showed survey data of US adults. So prior to the pandemic starting in 2020, about 11% of people surveyed reported symptoms consistent with anxiety or depression. Since that time, as of December 2020, that number went from 11% to 42%. I don't have the data up here, but since even 2020, those numbers have continued to rise, such that about 1 in 2 people, if surveyed, would probably wind up having symptoms of anxiety or depression. And it really seems like mental health has worsened for everyone, and across not just mild types of mental health conditions, but even more severe types. We've actually seen in some studies as much as a 7-fold increase in severe depression compared to mild or moderate. Some of the highest risk groups that we're seeing are women, healthcare workers, people from lower socioeconomic backgrounds, and people with pre-existing conditions. In thinking about how mental health has deteriorated in pregnancy, this definitely translates. We're definitely seeing that it's worsening overall. Similar to the numbers that we saw in the general population, in this particular study, they looked at pregnant people pre-pandemic and peri-pandemic and found, compared to baseline rates of anxiety and depression, which are around 20% for anxiety and around 10% for depression. In the pandemic, among pregnant people in this study, they found 60% of people actually had symptoms consistent with anxiety and 40% had symptoms consistent with depression. So we are definitely in an era where these are major issues for our patients. Ian Gottlieb and his group in the psychology department here in Stanford actually conducted a local study examining depression in our population of people here in the Bay Area, and then also sent a survey out nationally to examine how COVID has affected mental health in pregnancy. And similar to all the other research, they also found an increase in EPDS positive scores. They interestingly also examined different types of adversity in this study. So they looked at objective adversity, which is how the pandemic has actually affected people, for example, causing someone to lose their job, and subjective adversity, which is how they responded to the bad things happening to them, such as having fear of losing their job or fear of the unknown. And they found that subjective adversity was more associated with depression than objective adversity. And they concluded that factors tied to social and economic inequality enhanced vulnerability to stress related to COVID. We're going to have a thread of resilience throughout this because it can get a little bit depressing to talk about how bad mental health has really gotten. So our symbol of that is going to be these tomatoes. So we'll come back to it. So that's just setting the stage for how prevalent mental health conditions are for everyone right now, and particularly for pregnant people. So now I want to talk about what that means on a biologic level. And I'm going to talk a lot about this causal diagram of thinking about how increased levels of stress interplay with increased levels of inflammation in the body, how that goes on to contribute to pregnancy complications, and how all of those things together can contribute to effects in the child, who then themselves may grow up to have high stress, high inflammation, pregnancy complications in a cycle that can just repeat over and over across generations. So to begin, we will talk a little bit about how stress and inflammation are related. And I keep putting stress in quotations because people often refer to this in two different ways. So there's certainly psychological stress, which is a measure related to anxiety or depression or your mental health burden overall. And then this biological measure of stress, which is a measure of inflammation and the cascades that come from it. And these two types of stress are definitely related, but also distinct. This is just one example of the biologic plausibility for how these things are related. In this figure, you can see on the left, they're showing increases in pro-inflammatory cytokines directly affecting levels of serotonin and tryptophan in the brain, which go on to produce melancholic symptoms, anxiety, fatigue, etc. But also notice that while there's psychological effects on the right side of the figure, there's also psychological stress on the left side of the figure. So it's both a cause and effect of this. So this is very complex, but they're certainly interrelated. And this has been shown in pregnancy as well. In this study, they looked at different levels of pro-inflammatory cytokines among people in pregnancy who didn't have any mental health conditions, compared them to people who had anxiety, and compared them to people who had anxiety and depression. And across all pro-inflammatory cytokines, you can see there were increases among people who had anxiety or anxiety and depression. Now, this study didn't use a depression-only control group, which I think would have also helped interpret it, but you can see it's actually pretty interesting how dramatic the increase is across the board for anxiety alone. And I just want to emphasize that because anxiety is more prevalent than depression. And if that is having such a dramatic effect on pro-inflammatory cytokines, I think we should probably be focusing a lot more on anxiety in pregnancy than we do. This has also been extended into this idea of socioeconomic stressors and kind of thinking about psychological stressors in a different way. socioeconomic stressors have also been associated with inflammation in pregnancy. Some examples of this include looking at lower parental socioeconomic status, higher maternal childhood adversity, and lower maternal subjective social standing. All in pregnancy have been associated with increases in inflammation. The figure on the right is a little busy, but what it's trying to show is that across 3 time points in pregnancy on the x-axis, that as a pregnant person's perception of their social standing in their community goes down, their inflammation z-score, or the mean amount of inflammation in their body, increases. So those examples just touch on a few examples of how pro-inflammatory cytokines have been mapped out in pregnancy, but there are a number of biomarkers of stress out there that are all very interesting and definitely beyond the scope of this, but cortisol is a big one that's been looked at. Epigenetics is a big area of research. And then telomere length is another very popular biomarker of stress that we've been focusing a lot on. So just to give our research a little more context, I'm going to just define telomeres quickly here. So what are telomeres? They are repetitive DNA sequences that cap the ends of our chromosomes to protect the coding region from damage over time. They shorten as cells divide until they reach a critical length that trigger your cells to undergo apoptosis or tell the cells to stop dividing and enter this state of cellular senescence. They are thought to reflect the cumulative effect of biologic stressors on the cell. So they're thought of as a type of biologic clock, which we'll talk more about. And in a number of studies, shorter telomeres have been associated with increases in morbidity and earlier death in adults. And I just want to mention that there is a lot of nuance to understanding telomere biology, which has definitely taken me like 2 years to really feel like I have a better grasp on. But the most important thing to know about them is that they're cell specific. So as you can imagine, every cell is going to be dividing at its own rate. So each cell is going to have its own specific telomere length. So it's important to think about what tissue you're studying when you're using this biomarker. There are a number of determinants of telomere length. As I mentioned, the more a cell divides, the shorter it could get, the more exposed the cell is to things like smoking or other diseases. There are some cells that have an enzyme that actually lengthens your telomeres, something called telomerase. But no matter what tissue type you look at, they pretty much all correlate with each other. And in general, as you age chronologically, your telomere length tends to shorten. Leukocyte telomeres are one of the main types that have been studied, probably because they're the easiest to obtain in humans because you can get them from just a blood draw as opposed to a biopsy. And when looking at leukocyte telomeres, most people agree that overall they seem to shorten by about 20 to 30 base pairs per year over someone's lifespan. So again, this brings it back to this idea of a biological clock, or this biomarker that reflects a cumulative amount of your cell's aging process. And the reason that we've been so interested in telomeres is, again, because they really have been linked with this idea of stress. And I put stress in quotes again because telomere shortening can be accelerated by increased psychological stress as well as inflammatory stress. So kind of goes both ways. This is an example I've talked about before in other presentations, so people have probably seen this one. It is the study that the researchers did on surgical interns where they compared their telomere shortening over the course of a year and compared it to college student controls, and found that the surgical interns had 6-fold increase in the shortening of their telomeres in a year compared to the controls. And when stratified by different work hours, they found that the residents who worked the most had the greatest degree of telomere shortening. So I think all clinicians listening to this, at first would be concerned hearing that, but I have our resilience tomato thread here because there is a way to reverse this process. So it's definitely not finite, but it's an interesting example. And since psychological stress and biological stress are associated with accelerating telomere shortening, telomeres have also started to be thought of as this biomarker that you could study to understand the biological effects of adversity, which is also extremely interesting and a whole other topic of conversation, but I'll put a couple examples here. This was one study where they looked at people who were all age matched of different racial and ethnic backgrounds, and examined telomere length based on whether they were above or below the poverty line in Detroit. And they found some interesting differences. Taking that even one step further, adverse childhood experiences have also been associated with shorter telomeres. Adverse childhood experiences are pretty commonly reported on in pediatrics, I think a little less so in obstetrics, although we're starting to think more about it now. They are these experiences, things that happen to people before they turn 18 that have been associated with poor health in adulthood. And there's a lot of different ways to ascertain whether someone's had adverse childhood experiences. Most often you get it from a 10-question survey. Yes or no, has this happened to you? Captures events like trauma in your past. And they've shown that higher ACE scores, like as I mentioned, have been associated with poor health. But in this one pilot study, they also showed that higher ACE scores were associated with shorter telomeres in non-depressed adults. And there's just a figure here from that paper. I think you'll see from some of these images of data on telomeres that there's a lot of variability in this biomarker. And I think you'll find that with a lot of biomarkers of stress. So it's, certainly the case for telomeres as well, but still interesting to think about. So as taking all of that as context, we're going to now apply that to some of the research that we've been doing on cellular aging in pregnancy. So bringing it back to our causal diagram, we talked about stress and inflammation. So now we're going to talk a little bit about inflammation and pregnancy complications, specifically shorter telomeres and pregnancy complications. And I want to acknowledge many people who may be listening to this right now here at Stanford who did great baseline work on understanding the normal immune clock of pregnancy. And I think, what we've learned in trying to understand how telomeres fit into this larger milieu of the immune system is, nothing really acts in isolation. And you also really need to understand normal in order to understand abnormal. So just giving a shout out to that great paper. But really, when it comes to leukocyte telomeres in pregnancy, there's very little that is known. But I do want to take this moment to acknowledge that there has been work done on telomeres in placentas and fetal amnion. So there's this idea that, I mentioned telomeres are thought of as a biologic clock, but some are postulating that they may also serve as a gestational clock for the placenta as well. In some studies, they've shown shorter telomeres have been found in placentas and membranes of pregnancies complicated by fetal growth restriction, preeclampsia, and even spontaneous preterm birth. And then in this one mouse model study, they actually sampled telomeres from both chorion and amnion over time and found that right before spontaneous labor occurs in mice, the proportion of short telomeres in the placenta suddenly increases. So perhaps thinking again of telomere shortening as this gestational clock. So I think there's going to be a lot more for that to come, so stay tuned. But what we wanted to focus on was how leukocyte telomere length in mom changes over pregnancy and whether or not this could be something that could be used in the future as a predictive biomarker for adverse outcomes, just given its unique interplay between psychosocial stress and inflammation. So kind of similar to the immune clock of pregnancy, we realize there's really nothing out there about leukocyte telomere length and how it might change in normal pregnancy. So in order to understand that, we worked with the Merck Dimes Prematurity Research Center here to just characterize what normally happens to this biomarker in an uncomplicated, healthy term pregnancy. We also correlated leukocyte telomere length from moms with perceived stress constructs that were obtained in the first trimester. And we found over the course of gestation, there wasn't really a significant change in leukocyte telomere length, which was good. And you can see an example of our data over on the right, just seeing that over time, there isn't a huge difference. We included people that were sampled postpartum. That's why you see gestational age going up to 50 weeks. But when looking at the perceived stress constructs, the one thing that popped out for us as an area of interest was poor sleep, that in particular seemed to be the main stress measure that was associated with shorter leukocyte telomere length. So kind of gave us an idea about an area for future research. So then, after characterizing telomeres in a normal pregnancy and establishing that we could study them, we realized that we really needed to understand the effect of pregnancy on telomere length, because trying to compare what we found in the first study to the literature wasn't very successful, because most of the literature is not describing it in reproductive age adults. It's usually describing it in younger or older people. So in order to try to understand whether pregnancy itself might affect the natural rate of telomere shortening, we conducted a prospective cohort study here at Stanford. We enrolled 46 pregnant and 30 non-pregnant people in this study, and our primary outcome was to try to understand the change in leukocyte telomere length between 2 time points, trying to estimate the length of a term gestation. Because psychosocial measures are so important in understanding telomere dynamics, we also measured a number of psychosocial variables as well between the 2 time points. So you can see our timeline below. You'll notice that the study, of course, started in January of 2020, before any of us could have predicted what was going to happen. But we began in January, and then we halted, and then we resumed. But at time 1, we enrolled pregnant people between 10 and 14 weeks and age matched them to non-pregnant people who presented to our GYN clinic. At time 1, we drew maternal blood for leukocyte telomere length and asked all participants to fill out a PHQ-9 survey, which is a type of depression survey. We don't use it very much in obstetrics because we use the EPDS, but it's validated in pregnant and non-pregnant people, which is why we chose that one. And then after COVID came out, we realized how important COVID was going to be to interpreting our cohort's stress level. We worked with Ian Gottlieb and his team to adapt their survey to send to our cohort as well. Then at time 2, about 7 to 8 months later, we obtained a second blood draw and a second PHQ-9 score for our cohort. For our pregnant people, we drew their blood on postpartum day 1 for telomeres. And then for our non-pregnant cohort, we asked them to come back at a time point that would roughly match the pregnant people. But as you can imagine, once COVID came up, we had to become much more flexible in the timing for our non-pregnant cohort in order to bring them back to Stanford for research. So similar to what we found in our first baseline pilot study, we did not find a significant difference in leukocyte telomere dynamics between pregnant and non-pregnant people over time. So this is just showing a histogram of the rate of change per month in base pairs, and showing the pregnant histogram on the right and the non-pregnant histogram on the left. You can see for the non-pregnant group, we found that their telomeres shortened by about 6 base pairs per 30 days, whereas the pregnant cohort shortened by about 0.6 per 30 days. These weren't statistically different between each other. We had differential loss to follow-up in the non-pregnant group, so definitely, that's kind of why the histograms look a little different. But in general, no dramatic differences between the groups, which was reassuring because we had actually hypothesized that pregnancy might accelerate shortening just because it's physically and biologically stressful. Kind of bringing this idea home that there's not a lot of change in this biomarker over the course of pregnancy. The telomere lengths for both our pregnant and non-pregnant participants were very highly correlated with the telomere length at time 2. And the reason why I think this is important to report is that, in other research using telomeres in pregnancy, which is very limited, they're drawn at very different times. And so I think it's important to know that from what we can find, that's probably okay. It's probably okay to draw it in the first trimester, postpartum day. They seem to correlate within individuals over the course of a year, which is nice for future research. Secondarily, in this cohort, we examined psychosocial measures, as I mentioned. And overall, we had a pretty low baseline prevalence of depression in our population. But even despite that, the majority of people reported moderate or severe worsening of stress levels related to COVID. And about 1/3 had worsening of their depression scores. The figure on the right just shows, probably as you could imagine, people who had stress levels that worsened moderately or significantly were more likely to have higher EPDS scores postpartum. We then conducted a subgroup analysis in this prospective cohort, thinking about this idea that we know inflammation in pregnancy is probably associated with some of these adverse outcomes, such as preeclampsia. and mental health, as I've already mentioned. So because telomeres are considered a biomarker of stress, we wanted to see if we could use first trimester telomere length as a predictive biomarker. So we took the 46 pregnant people that we had in our cohort and examined their first trimester leukocyte telomere length to see if it could potentially predict anything that happened in these pregnancies. We did find that shorter first trimester maternal leukocyte telomere length correlated significantly with earlier gestational age at delivery. And again, this is a small cohort. This is a subgroup analysis, definitely not powered for this, but you can see we did find a moderate correlation. Definitely is worth looking at in larger cohorts. We also looked at some other specific pregnancy outcomes that are thought to be related to inflammation, such as hypertensive disorders and spontaneous preterm birth specifically. And as I mentioned, this is a prospective cohort, definitely not powered for any of these things. We just wanted to explore these. But we did find that the first trimester leukocyte telomere lengths were shorter in people who went on to develop hypertensive disorders and spontaneous preterm birth. But as you can see, we only had 3 spontaneous preterm births in the whole cohort. So definitely exploratory, but needs more work. And this is just that same data shown graphically. So you can get a sense of the distribution of the telomeres. We then took that one step further in a poster that we just presented at SMFM, trying to do a nested case control study to better understand what we were finding in the prospective cohort. We did this in partnership with the March of Dimes Prematurity Research Center, as well as the Boston Birth Cohort and Johns Hopkins. I put this table here just so you can get a sense of why we used both of these cohorts, because we felt that they told us interesting stories based on their demographic differences. And as you can see, again, these are very small studies. All of this is pilot data, but we did find trend towards shorter telomeres in people with preeclampsia, mainly in cohort 2, not so much in the Stanford cohort. So we're still trying to work on this and sort of integrating this with other stress measures to see what the next steps are there. But this is certainly the iceberg, tip of the iceberg, if you will. There's a lot more to come and a lot more we're working on there that hasn't been presented yet, but I think there's hopefully going to be more to come from that. So just in the sort of last few minutes, I want to talk about the transgenerational part of this, which we haven't quite yet had a chance to incorporate in our studies, but I'm hoping we can in the future. And that's really just how this stress environment affects the offspring, and kind of bringing it back to the tomato example. Like I said at the beginning, I think we all probably fundamentally know that a stressful environment probably would affect your offspring. And there's no reason to believe that would be any different for tomatoes as it is for humans. But bringing it back to our causal diagram, we've talked about stress, inflammation, pregnancy complications. There's no reason why all of those things wouldn't affect the child. And it does seem that in research trying to examine this, they have demonstrated that there are some of these transgenerational effects of stress. So it's sort of unclear, is it that stress and inflammation predisposes a person to pregnancy complications, and those complications have downstream effects for babies? Is it that a pro-inflammatory environment itself affects fetal development? Is it a little bit of both? Hard to say. The fetal programming idea has been described for a number of biomarkers. It has also been described for telomere biology, specifically thinking about how fetal programming can affect a neonate's telomeres and how that might have impacts for them across their lifespan and sort of what that reflects. So there have been a number of studies looking at fetal determinants of telomere length in babies and children. And they have found, sort of along a similar vein, that there are a number of stress measures that are associated with shorter neonatal telomere length. For example, this study here looked at pregnancy-related psychosocial stress and found that as that increased, a baby's cord blood telomere length decreased. You can see again here, there's a lot of variability in telomeres, so it's definitely something to keep in mind when you study them. But we tried to look at some cord blood in one of our studies and found a similar degree of variability. And I think it's probably hard to ensure you're not having maternal contamination, which is a main problem when you're looking at leukocyte telomeres. So I think that's the part of the neonatal piece that we need to work on for our research. An effect of inflammation on neonatal telomere length has been shown not just for stress, psychosocial stress, but also for inflammatory stress. So this study was looking at as maternal inflammation went up in pregnancy, newborn leukocyte telomere length went down. So there's certainly some sort of transgenerational field programming pathology going on. And then this is just the last one that I always like to show, because this shows that it's not just the neonatal telomere length that's affected, but this study actually went one step further and examined telomere length in children aged 3 to 5 years, and found that as their mother's psychosocial stress score in pregnancy went up, the child's telomere length at age 3 to 5 years went down. And I highlight the two slopes that are related to the second and third trimester stress, because again, we emphasize so much on postpartum mental health, which is extremely important. But I think mental health in pregnancy and trying to intervene in pregnancy is what potentially could be, the intervention that breaks the cycle. So again, we've had a very surface level look through this proposed causal diagram. I think there's a lot more to come, and a lot of people who are working on this here at Stanford, as well as at other institutions. So I think it'll be pretty interesting stuff coming. But I mentioned we would end on a silver lining, because thinking about this cycle as sort of a never ending can be depressing. So it's important to remember that it's not necessarily just the stress itself, but perhaps how you respond to it that matters, kind of bringing us to our tomato analogy. And I bring it back to telomeres again, because this biomarker has also been shown as sort of a resilience biomarker too, which is why it's so unique, I think. So they have shown that different lifestyle modifications can affect the rate of telomere shortening. Stress reduction, diet changes, smoking cessation, physical activity, sleep, all these things have been shown in different studies to be of benefit on a biologic level. This is just one example of a study showing the interaction between physical activity and stress on telomere length. Of course, not in pregnancy, because we don't have enough in pregnancy yet, but at least outside of pregnancy, at the top of this graph, you can see among sedentary people, the higher their level of stress, the higher the probability of them having short telomeres. But among active people, you don't see the same association. So definitely gives some hope for future interventions that might have an effect biologically. And this has been touched on a little bit in pregnancy, not so much yet, but I think this is just an interesting study, definitely worth a read, where they actually looked at not maternal stress in pregnancy, but actually a resilience score, which was the first time I had read about that. But as a pregnant person's resilience score, or ability to be resilient against stress, increased, their child's telomere length increased as well. So, I think it's just showing that these things can change a little bit. And so gives us some areas to think about interventions. I think trying to come up with interventions that our populations might actually do. And if we can show biologic changes with these interventions, I think that would be pretty cool. So in conclusion, the last 2 years have been stressful, definitely an understatement. Prenatal depression and anxiety have increased, and social and economic inequalities have enhanced vulnerability to stress. This is important because stress can affect pregnancy on a biologic level, and I would posit it's not just stress, anxiety in particular, I think is a big thing we need to be studying more. Stress biomarkers may be associated with pregnancy outcomes that have effects across the lifespan, and perhaps leveraging resilience through interventions is the key, we hope. We'll see. So we have a lot of areas of investigation happening right now in this umbrella, if you will. So we're continuing to look more at telomere dynamics in pregnancy. I have some of the authors that are working on this with me. So thank you to everyone here. This is definitely not an exhaustive list, but we're trying to understand some baseline wellness trends in pregnancy. I mentioned sleep seems to be a big factor. That's sleep and physical activity, both that are tied with telomere length. So trying to see if we can come up with an intervention that we could track biologically would be pretty cool. We also have some work in general looking at mental health and pregnancy outcomes. And part of my time now is trying to come up with interventional trials to use all this baseline data to see if we can actually make an impact. So I like to end just on some positive pictures here, thinking about our resilience. I think, we're all very used to Zoom now, but I think this picture of audio only versus with video makes me laugh every time. And I think it's very fitting to give this talk during ACOG Wellness Week. So just kind of think about all the positives that you have to kind of counteract the negatives. A big thank you to many of my mentors listed here. This is definitely not an exhaustive list. Thank you to the Dunlevie Center for having me today to talk about the work that we're doing. I also want to specifically thank Stanford MCHRI for funding a lot of this research and also funding a master's in epi that I'm doing right now. So thank you all so much. I'm happy to take any questions. And if you are interested in working on any of this, please email me. We are happy to collaborate. So thank you all.
Danielle, that was a fantastic talk. Anna, do you want to run through the questions that are in the chat?
Sure. So Dr. Peterson is asking, do you know if there is any data on lymphocyte telomere length where women are undergoing anti-inflammatory treatment, for example, low dose naltrexone during their pregnancy?
I think it's a really interesting question and something that I want to understand better. And at this time, no, but that's really because there's just not a lot about pregnancy in general in terms of telomere dynamics. So I think that, another big one that comes to mind is aspirin use. That alone is something that we're trying to look at in our prospective cohort again. But I think that that's going to be an interesting biomarker to study the effects of anti-inflammatories, not in isolation probably, but with other biomarkers.
Great. And Dr. Bianco is commenting that steroid use during pregnancy is also a big one.
Yeah, that also, we didn't have too many people in our study who got betamethasone during the prospective cohort at least. And a lot of our other research has been in control healthy pregnancies where nobody got steroid use, but anything that can affect your leukocytes, can theoretically affect their rate of division. So these are all things that we have to think about for sure in future research. So yeah, especially as we start thinking more about pregnancy complications, that's going to be a big part of it.
Great. And Dr. Lyle had a comment and question. Thanks for an interesting talk. Did the study on pregnancy stress and childhood telomere length correct for early childhood / household stress? Was stress during pregnancy a correlation with their children's telomere length or causation?
It's a great question. And I think that is the root of the issue, is that we can probably never disentangle the causation versus association piece of this. I think especially when you think about disparities and especially when you think about childhood telomere length, there are probably many other things that factor in to it. And the other piece of that is certainly, is it pregnancy that's doing any of this, or is it being a parent and how your child's being raised? For the study looking at childhood telomere length, absolutely, if a woman is more stressed during pregnancy, does that mean she's more stressed in the 5 years raising the child? And does the conditions in which the child's raised contribute to their telomeres? Certainly, those things are all probably happening together.
Danielle,
you did not look at the early childhood or household stress in your study?
We did not, but I think we are working on future stuff looking at ACEs and things like that with telomeres. So that's our hope.
Danielle, I thought your last bullet was especially important, the issue on resilience. The mind can make heaven hell and hell heaven, and trying to objectify some of this idea of stress. One, have you ever thought of having some international partners, going to areas where there could be refugee camps, war zone, things that we would clearly know as profoundly stressful, and testing this theory in, across those different landscapes?
Yeah, I think that would be a really cool future area. There's some people that have looked at telomeres in some different international countries, trying to look at the effect of nutrition. There's one study in Rwanda looking at childhood nutrition. So I think there's definitely areas for that. And there's a big group out of Berlin actually that's really interested in stress and pregnancy that we've been in touch with too. So I think that would be great.
Yeah, I was just typing a comment about resilience, yeah, sir, when you were asked your question. So I was just curious, do you have more information about how the Werner article measured maternal resilience?
So they developed it. There was a resilience questionnaire basically that had been developed that then they took and adjusted and then used a score on this questionnaire. But it's kind of, I think, we're just starting to think about this, and I'm sure there's a question of, is that the best way to estimate it? How else could you estimate it? But it's interesting to think about, what is the best way to measure that? Good question.
Yeah, great. Dr. Nguyen had a question. Given tissue differences in telomere length, do you think that different types of leukocytes will have different lengths? And are you looking at that?
Yes, Dr. Nguyen. I know she and I spoke about this, but there are ways that you can. So all of our research is basically taking all of your white blood cells and averaging their telomere lengths, which has its pros and cons. There are many different ways to analyze this biomarker. And there are ways using flowfish, for example, that you can look at different subsets of leukocytes, and each of their telomere lengths can be pretty granular. So we're trying to figure out if we need to do that, or how much that's going to tell us that's different from what the sort of mean measure is telling us alone. And I think there's pros and cons to both, and certainly something to look at. And it does seem that there are different subsets of leukocytes that there's variation between them. So it's definitely a thing to think about.
Wonderful talk, Danielle. Thank you so much. We are so proud of you and all the work that you are doing, and look forward to hearing from you again in the near future.
Thanks for having me.
Thank you all for joining.
Great. Thank you all. Until next time, stay safe.